Hello everyone, in this video I'll be explaining on the application and conclusion of fluorescent microscope. This fluorescent microscope is an optical microscope that uses fluorescence and phosphorescence instead of or in addition to scattering, reflection and attenuation or absorption to study the properties of organic and inorganic substances. Fluorescence is a phenomenon that occurs when an object is impacted by a given wavelength of light and emits light of a wavelength longer than the one to which it was exposed. There are two types that are fluorochromine and immunofluorescence. Fluorochromine that involves the non-specific staining of any bacterial cell with any fluorescent dye, while immunofluorescence uses antibodies labeled with fluorescent dye to specifically stain a particular bacterial species. The uses of this fluorescent microscope includes fluorescent microscope has also been employed for detection of antigen and antibodies. It also used to identify structures in fixed and live biological samples. The advantages of this fluorescent microscope includes fluorescent microscopy is the most popular method for studying the dynamic behavior exhibited in live cell imaging. This stems from its ability to isolate individual proteins with a high degree of specificity amidst non-fluorescent material. The sensitivity is high enough to detect as few as 50 molecules per cubic micrometer. Different molecules can now be stained with different color, allowing multiple types of the molecule to be tracked simultaneously. This factor combined to give fluorescent microscope a clear advantage over other optical imaging techniques for both in vitro and in vivo imaging. Next, coming to the limitations of fluorescent microscope, it includes fluorophores lose their ability to fluorescence as they are eliminated in a process called photobleaching. Photobleaching occurs as the fluorescence molecule accumulate chemical damage from the electrons excited during fluorescence. Cells are susceptible to phototoxicity, particularly with short wavelength light. Furthermore, fluorescent molecules have a tendency to generate reactive chemical species when under illumination, which enhances the phototoxic effect. Unlike transmitted and reflected light microscopy techniques, fluorescent microscopy only allows observation of the specific structures which have been labeled for fluorescence. So this is the application, advantages and limitations of fluorescent microscope.